Hello friends, how are you? Hope all are fine. Friends, today we shall discuss about the mechanism of nucleotide excision repair. Before that, I request all of my friends, please subscribe my YouTube channel and watch, like and share my videos with your friends. Nucleotide excision repair (NER) is a versatile DNA repair system that removes bulky and helix distressing lesions. This pathway is elaborate and includes two distinct mechanisms such as global genome repair, GGR, and transcription coupled repair TCR. Global genome repair is responsible for repair of the entire genome, whereas TCR preferentially repairs transcribing DNA strands. The basic steps of nucleotide excision repair are: number 1, recognition of the damage. This is followed by unwinding of double-stranded DNA, in the neighborhood of the damage. Third step is excision. Of the damaged nucleotides. And finally, DNA polymerase and ligase fill the gap by DNA synthesis and ligation. In humans, nucleotide excision repair is a major defense against two carcinogens such as UV radiation and tobacco smoke. Defects in the human NER is associated with genetically inherited diseases such as xeroderma pigmentosum, Cockayne syndromes, and trichothiodystrophy. This figure illustrates the mechanism of nucleotide excision repair of pyrimidine dimers. This is a pyrimidine dimer. Pyrimidine dimers are molecular lesions originating from thymine or cytosine residues within the DNA. Such lesions are formed when DNA is exposed to UV light, particularly UVC, that result in the formation of covalent bond between adjacent thymine or cytosine bases. This figure shows a thymine dimer in which adjacent thymines are linked by covalent bond. In E. coli, nucleotide excision repair of pyrimidine dimer is carried out in a ATP-dependent process through the action of three proteins such as UBR-A, UBR-B and UBR-C. This system, which is referred to as UBR-ABC endonuclease, UBR-ABC endonuclease that cleave the damaged DNA strand at the 7th and 3rd or 4th phosphodiester bond from the 5' and 3' sites respectively. In this figure, this is a pyrimidine dimer which has been formed due to the exposure to UV light. The system known as UBR-ABC endonuclease cleave the damaged DNA strand at the 7th and 3rd or 4th phosphodiester bond from the 5' and 3' sites respectively. In the next step, the 11 or 12 nucleotide segment is displaced by binding with another protein called UBRD or helicase 2. The excision and removal of this oligonucleotide results in a gap which is then filled through the action of DNA polymerase I, which adds successive nucleotides to the free 3-hydroxyl group in 5' to 3' direction. And finally, the nick which is left behind is sealed by the action of DNA ligase. In this way, various helix-dispersing molecular lesions within the DNA are corrected by nucleotide excision repair, thereby restoring the integrity of the genome. Friends, for more updates, subscribe my YouTube channel, Sam Biology Class.